We're so hands-on, and we do the same research that a lot of professionals do.
It's very important to know that as a high school student, I have the opportunity to be involved in real research.
Yeah. It's not many high school kids who get to do this.
Got it.
Why don't you put some DNA in?
Okay. Sure.
Most of science I've learned has always been in the classroom, and I was never very interested because all you did was really memorize a bunch of facts, but here you apply it.
You get that?
What are you doing?
We're doing
PCR.
PCR.
What does that mean?
It's a very effective way to amplify the amount of DNA we have. Should be enough here.
The ITEST project is a genomic sequencing project. Students, are going to sequence cDNAs from brine shrimp Artemia franciscana. The program was developed to try to introduce teachers and students to what's actually happening in research. And their motto has always been, to learn science by actually doing science.
Why are you doing a digest mix? Like, why not just do it individually?
Well, because it saves a lot of time as opposed to individually making one by one.
And the quantities are larger, so it has less error.
Okay, good. The way that we do in East Brunswick is we have it as an after-school class. It's period 13. What I wanted to do with the students is to make it fun for them. That was something that they can be actively engaged in and something that they want to continue in. Because if there's something that we can turn them on to, I think that's really important, especially in research. Everything that you compare it to,
Control.
Okay, so you got your control. All right,
Everyone looks forward to coming here because it's just something so different from anything that anyone has ever done in our age group.
And we've all become friends after working together.
Okay, is that 3?
No, that's 2.
That's 3.
That's 2.
The students come into the project and they get a Petri plate with cells on it, and each of those cells contains a fragment of DNA from the brine shrimp that's different. So every colony or every cell that they pick contains a different DNA fragment.
Now the next step is the mini prep. So can you explain to us what's going on with the mini prep?
All right, so basically what a mini prep is that we're separating the plasmid DNA from the bacteria.
Do you have your balance tube?
Yeah.
We have our plasmid, which is a part of the bacteria, and we perform different lab procedures on them. We do restriction digest, we do PCR, and we run gels, and we separate the actual DNA of the Artemia that we're trying to find.
You want to adjust that a little bit?
The students are doing the same exact techniques and using much of the same equipment that I use in my own research lab. One of the goals that we have is to try to get the students involved in research early and that help them continue on throughout their college career. During the summer, teachers from roughly 20 local high schools come to Rutgers University and they learn some of the background in terms of microbiology, genetics, and bioinformatics. And they also learn how to conduct the experiments that they're going to need to use for the project.
I feel that I'm more in tune with what's going on in my field, and I feel that I can be a better teacher to be able to bring this information back to my students. It was an opportunity for them to find out what research was about, and they might consider that as an alternative in their career choices. So I think that is invaluable.
GE Healthcare is involved with the project because they have a very strong commitment to community outreach. They've developed a mentor program where GE employees actually talk to the students about career options in the sciences from industry side.
So when you think about entering science, ideally think about longer term what it is you want to achieve.
GE has an interest in educating students in science, and hopefully some of these students will come back and work for GE Healthcare in the future too.
All right, one of the things that's important is to analyze your DNA. This is where the hard research comes in.
The students can begin to perform a bioinformatics analysis to analyze their DNA sequences. And that's where really a lot of the discovery occurs.
That's the P-X. Yeah.
It's a very creative process. You're just looking at a bunch of letters. And like from these letters, they start seeing that these letters take on a structure. And they start looking at the structure of the protein. And I think that makes them look at larger pictures. And just their level of thinking is way beyond where they would have been as a normal senior in high school.
So what kind of matches are you getting right now?
You never know what you're going to get. It's almost like a lottery because you don't know if you're going to win or not. You don't know if you're going to get some, discovery or not.
The students eventually get to publish their DNA sequences up on the databases at NCBI, the National Center for Biotechnology Information. And so as a result of going through this program, they become published authors.
Most high school students will never have the chance to be published. The sequences that we found, they're out there on the web for any scientist to use. And that's just an amazing thought. Maybe somewhere down the road, a sequence that I found will help create a cure for disease.
SMBC may have benefits to Alzheimer's disease.
Over 200 students come to GE Healthcare with their teachers and their parents, and they're presenting the results of their research. And GE Healthcare scientists are actually talking to them about their research, asking them questions about what they've done.
I find it really enjoyable when the students get it, or they come back and ask a question about, gee, how does this work, or why does this happen, or if they make an observation that I may have missed. And so that's really exciting.
I think it's one of the best things I've ever done in my career because I've learned so much and I've been able to bring so much back to my students that I would never have been able to do without this program.
It's kind of amazing that we're doing the same stuff that people in college are doing, people in grad school are doing. We're using the same programs, same resources. And, you know, it feels like we're a part of something that's really big and really important.